Hi, everybody. My name is Ed Davis. I'm Senior Application Scientist at GeneCopoeia Incorporated, located in Rockville, Maryland, just outside of Washington, D.C. I want to thank you all for joining us for today's webinar entitled Harnessing CRISPR for Activation of Gene Expression. We really appreciate it. Before I get started, I just want to go over a few practical matters. First, you may ask me questions at any time. But to do so, you won't be able to speak vocally to me because your microphones will be muted. Instead, you'll need to type your questions in the questions box in your control panel. If you can't see your control panel, it might be hidden, in which case look for a small strip of buttons on one side of the screen. One of those buttons will be orange with a white arrow, and when you click on that button, you will then be able to see your control panel and hence the questions box. Secondly, I'm going, in the interest of keeping the talk going smoothly, I'm going to wait till the end of the talk to answer your questions in the order in which they were received. And then I will read each question out loud as well as the answer out loud for everybody to hear. And with that, let's get started. Okay, so first I want to tell you who we are at GeneCopoeia and what we're about. We were founded back in the late 1990s as a provider of genome-wide collections of human and mouse open reading frame or ORF clones. These are ready to express plasmid clones, mostly for cell culture, but they can also be used for bacterial expression or in insect systems. But the whole point is that they are ready to express in the cell system of your choice. And since then, we've expanded out into a number of other clone types as well as other types of reagents. So we have clones for, as I mentioned, ORF, promoter, microRNAs, CRISPR, of course, and shRNA, as well as a few others. We also provide products and services for viral systems, primarily lentivirus and AAV, where we make either, in some case, for at least in the case of lentivirus, packaging kits, and for both lentivirus and AAV, we provide ready-to-use particles, infectious particles. We have a number of stable cell line products, including CRISPR-Cas9 stable cell lines, labeled cancer cell lines, and cancer biomarker cell lines. We also have large collections of kits and reagents for transfection, luciferase, fish probes, indel detection, and cloning. And what's not on here, I just realized we also have qPCR kits, and that's very important. And finally, we have a number of solutions for fluorescent detection, including cell function assays to assay apoptosis, for example, nucleic acid detection for doing quantification for next-gen sequencing or gel electrophoresis, cell structure probes, and fluorescent dyes. But of course, you're here to learn about a CRISPR-related topic, so let's talk about that. Here's an outline of today's seminar. First, I'm going to give you a brief overview of CRISPR technologies and applications, then an introduction to CRISPR activation, which is, of course, the focus of today's webinar. Next, I'm going to talk about ways in which to use these reagents. So I'm going to talk about delivery methods as well as preparation and screening methods. So let's talk about CRISPR. So CRISPR, of course, has been a very hot technology for about the past 5, going on almost 6 years now. Its interest has not waned at all over the past several years. And it's been widely used in many types of systems, from lower organisms, including bacteria and yeast, all the way up to mammalian systems. And there are gene therapy trials going on. And it's all based on this relatively modest adaptive immune system that's found in most bacteria and archaea. This is a figure from an old paper from Feng Zhang's lab showing the original CRISPR system, or the most widely used CRISPR system, which is based on a Cas9 nuclease shown represented by the yellow shape from the bacterium Streptococcus pyogenes. This is actually a 3-component system where the Cas9 nuclease complexes with 2 RNA species known as guide RNA, CRISPR RNA, and tracer RNA, which can be combined to form a single guide RNA. When these combine into a complex, the Cas9 nuclease is transported to a target site represented here in blue that can be programmed into the RNA. This target site, when found in the genome upstream of a 3-nucleotide site that consists primarily of NGG, but it can also be NAG. This is also known as a protospacer adjacent motif or PAM site. When the Cas9 nuclease is guided to this sequence in chromosomes, it makes a double-strand break, which can then be used for many applications, from gene knockouts to knocking in transgenes or making base pair changes or correcting base pair mutations, etc. So this is the most widely used version of the CRISPR system. There have also been a number of variants and improvements made on it since. And so this table shows some of those variants. So the first row shows the SP-Cas9, as the Streptococcus pyogenes Cas9 is known. This is the unmodified Cas9. But there have been a number of variations on this, such as a mutant that was recently created of SP-Cas9 called SP-Cas9NG, which relaxes the PAM requirement to NG, so it gives you more target site flexibility. There have also been a number of mutants created in SP-Cas9, known as SP-Cas9HF or ESP-Cas9, which are intended for giving higher fidelity and specificity to the CRISPR system. I'm not really going to go into this today. It's beyond the scope of the webinar, but these variants do reduce off-targeting. There's also a new version called Hi-Fi Cas9, which also reduces off-targeting. And then there's versions of the CRISPR system for other organisms, such as CPF1, which is a nuclease found in 2 different organisms. It's smaller than Cas9, SP-Cas9, and it's simpler. There's no requirement for a tracer, RNA, etc. This is just an alternative version. There's also Staph aureus Cas9, which is smaller than SP-Cas9, so it's usually used with adeno-associated virus or AAV. And then there's dCas9, which stands for dead Cas9. This is going to be very important for today's talk. It's a nuclease dead version of Cas9 for tethering to effectors. And I'll talk a little bit more about that in a minute. And finally, there's Cas13, which is a completely different animal, completely different kind of Cas enzyme from this species, Elwadia. It targets RNA, so it actually edits and manipulates RNA. So now let's talk about CRISPR activation, because of course that's the focus of the talk today. So the CRISPR system has been adapted for not just gene editing, that is making permanent modifications to the genome like the Cas9 nuclease does, but it's also been adapted for either activating or repressing genes. So there's a whole history of this for other systems, like you may have heard about TALEN or zinc fingers. Those are older gene editing technologies that have been used to tether not just to DNA endonucleases, but also to transcriptional effectors like repressors and activators. Well, the CRISPR-Cas9 system is no exception. So the dCas9 that I mentioned before has also been targeted to transcriptional effectors. Again, these can be for repression or activation. Repression would be similar in many ways, at least functionally, to shRNA for knocking genes down. But transcriptional activation is something completely different. And again, this is all dependent on, at least for CRISPR purposes, on this nuclease dead Cas9. Okay, so the crystal structure of the nuclease dead Cas9 complexed with sgRNA is shown here, also from Feng Zhang's lab. And this is important because the Cas9 nuclease has in its wild type form, has 2 domains, a RevC domain shown here in cyan and an HNH domain shown here in pink. So what researchers did a number of years ago was they mutated one site in each domain. One is a aspartate to alanine substitution at position 10, and the other is a histidine to alanine substitution at position 840. That's in the HNH domain. So what this does is this completely knocks out the nuclease function of Cas9, but it is still structurally sound, and it is still able to be targeted to chromosomal sites by single guide RNAs. So now again, we're talking about CRISPR activation. This is an important area of research because not only can you do gene ablation studies to interrogate the function of genes, but you can also do overexpression studies, which is something that's not as commonly used, but it's very important. So why use CRISPR for this? I posed this question in the slide. So you can, of course, overexpress genes in other ways. The most widely used method, of course, has been to use open reading frame clones or ORF clones, and that's something that we at GeneCopoeia specialize heavily in, is these open reading frame clones for overexpression. But ORF clones, of course, you tend to use artificial promoters like CMV or EF1 alpha, which is fine in most cases, but in some cases it's not. It depends on what you're studying. Another issue with open reading frame clones is not all protein isoforms are always covered. So you might have to express several different ORF clones to cover all the protein isoforms of a particular gene. Also, large ORFs are sometimes too difficult to clone, so there can be ORFs that are 8, 9, 10, or even more kb. So those can be difficult to clone into plasmids. And of course, having to express multiple ORFs for a given gene also makes it difficult for carrying out and cumbersome for carrying out large-scale screens for like using libraries, for example. So CRISPR has been adapted to alleviate most, if not all, of these limitations of other methods of doing activation. So now again, there's been a history of this going back several years, but I'm really just going to focus on the most widely used and most successful method for CRISPR activation, which came also from Feng Zhang's lab, this paper from Konerman et al., going on almost 4 years now. This is known as the dCas9 SAM system. SAM stands for Synergistic Activation Mediator. Okay, so this dCas9 SAM system is a 3-component system. Component number 1 is this dCas9, this nuclease dead Cas9 that's fused at the protein level. This is a fusion protein with the VP64 transactivator, transcriptional activator from herpes simplex virus. Okay. Now, of course, in order to get this complex to genome sites, you need a single guide RNA. That's component number 2. But the sgRNA has been modified in this system in as follows. So you could use Cas9, dCas9, VP64 by itself for transcriptional activation. That's what was done in early iterations of this technology. But in most cases, the activation has been either weak or non-existent. So what the Konerman et al. Group did was they set out to improve this system. And so by doing several different hypotheses and testing, they came up with a system where they modified the sgRNA to contain an aptamer that binds to the protein MS2, which is a bacteriophage coat protein. Okay. And through further testing, they were able to fuse the MS2 protein with the P65 protein, which is a transactivating subunit from NF-kappa B, as well as HSF1, which is an activation domain from human heat shock factor 1. So and this is known as MSH, okay, this fusion protein. This is a fusion protein. So both the MSH fusion protein and the VP64 protein recruit overlapping but distinct sets of transcription factors. Okay, so the idea is that once you get this complex, you will recruit a whole host of transcription factors to whichever promoter region this sgRNA is targeted to. Okay. Now, one of the nice things about this system is it harbors, it still retains the simplicity of the Cas9 nuclease system, in that you can use the same type of 20 nucleotide sgRNA sequence that recognizes NGG PAMs as the Cas9 nuclease does for DSBs. And what they also found, importantly, and this was a nice finding, is that optimal activation occurs with sgRNAs that are targeted anywhere from up to 200 base pairs upstream of the transcription start site. So that made the design rule fairly simple, actually. So you could design multiple, let's say you want to activate a particular gene in a cell, you could design multiple sgRNAs that target the promoter, and then you could test them and then just pick the best one. Okay. Whereas with the dCas9 VP64 alone system, again, you could use that for gene activation, but what people tended to have to do is they would have to use multiple sgRNAs at one time synergistically, and that would increase the complexity. But with this, you can get away with just one sgRNA. Okay, so when the Kuhneman et al. Group made this system, they found robust activation for many protein coding genes and even for a few long intergenic non-coding RNAs, also known as link RNAs. The data, which is shown here, they found this shows sgRNAs that they tested fold activation for sgRNAs at different positions upstream of the transcription start site. So in most cases, they got several fold activation, like for VEGF-A, they got 4 to 5 fold activation. But they also got activation for that would even be tens or hundreds or even thousand-fold above basal level. So this gave a quite robust activation system that was anywhere from 15 to 20-fold more efficient than the dCas9 VP64 alone. Okay, so now that I've told you about that, let's talk about how to use this in an experiment. Okay, so I'm going to focus mainly on experiments that are designed for cell culture. Of course, you could probably get this. You could get this to work in transgenic animals, but I'm going to just talk mainly about delivery methods, because GeneCopoeia has a number of solutions and products that are useful for these methods. Okay, so the first delivery method I want to talk about is plasmid DNA transfection. Okay, so this shows kind of a generic type of plasmid where we could have the dCas9 VP64 as well as the aptamer modified sgRNA on a single plasmid. Okay, this would be kind of an all-in-one plasmid, but you would also need a separate plasmid or possibly even the same plasmid expressing the HSF P65MS2 fusion. These plasmids provide fluorescent reporters, which can permit sorting, which can be very useful, of course. And selectable markers are available for stable integration if desired. But now this can work very well, but Cas9 needs to be transcribed and translated. Okay. So the second delivery method I wanted to mention is to use RNA transfection. Okay. So with RNA transfection, you would co-transfect in vitro transcribed dCas9 VP64 mRNA, MSH fusion mRNA, and sgRNA, the adapter modified sgRNA. And the sgRNA can be either encoded on a plasmid, in vitro transcribed, or synthetic. Now, the advantage of using RNA is that it's plasmid-free, so it can't integrate. And sometimes integration that you can get with plasmid transfection can be deleterious. But in this case, the dCas9 VP64, as well as the MSH, needs to be translated. Still needs to be translated. The third delivery method I wanted to mention is protein transfection. Okay, so in this case, you would preform a complex of the dCas9 VP64 protein with the aptamer-modified sgRNA in vitro to form an RNP. And the aptamer-modified sgRNA can be either chemically synthesized or in vitro transcribed. And then you would transfect the cells with this complex using either lipid transfection reagent or electroporation, and you would co-transfect with the MSH2 mRNA. And this is a great method because it does not integrate, unlike plasmids. Okay, so those three methods I just mentioned were all transfection-based, and so I wanted to mention that it can work with most transfection reagents, but GeneCopoeia has a transfection reagent known as Endofectin Max, robust transfection reagent. And this is our own brand, of course, of transfection reagent and it gives equal to or greater transfection efficiency compared with lipofectamine 2000 or lipofectamine 3000, as well as a few others like Fugene. It's especially robust with difficult to transfect cell lines like neuronal cell lines. We've had customers test this out and they've gotten very good results. And it's also capable of efficient transfection of DNA, RNA, as well as protein. Okay, so it gives it's a very robust transfection reagent, and it works great with CRISPR methods, including CRISPR activation. Okay, and the fourth and final delivery method I wanted to mention is lentivirus. Okay, so why lentivirus? This is a completely different way of delivering components into your cells. Well, DNA transfection is not always possible or practical. Some cell lines are difficult or impossible to transfect. Yet most mammalian cells support infection by engineered lentivirus. So sometimes it's very much desirable to use lentivirus, but the one caveat with lentivirus is it by default integrates into the genome. So your experiments would have to be would not have to be compromised by that kind of integration. But it is a very robust method for delivering these reagents. And I mentioned lentivirus as a way of introducing yet another GeneCopoeia product that's specific for gene activation, and that's our GeneHero Cas9A or Cas9 activation stable cell lines. So these are cell lines with the dCas9 VP64 and the MSH stably integrated in the genome. Now they're integrated in different places because we use lentivirus technology to integrate these transgenes. So the dCas9 VP64, driven by a heterologous promoter, is integrated on one chromosome, and the MSH, driven by another heterologous promoter, is integrated on another chromosome. We currently have two pre-made cell lines available for HEC293 and MCF7, and these are functionally validated for CRISPR-mediated activation. They're ideal for lentiviral CRISPR activation applications, but they can also be used for non-viral CRISPR applications as well. And then you would just need to provide sgRNA, aptamer-modified sgRNA for that. So this is the performance data for our CRISPR-A cell lines. This is the HEC293 cell line. This shows activation data for 3 different genes, as I meant. So these are the qPCR curves for VGFA, EGFR, and HBG genes. The green curve represents cells transfected with aptamer-modified sgRNA, and the blue curve is the no sgRNA control. And in all 3 cases, we get very robust CT curves and very robust application. All of these are over anywhere from 16 to almost 50-fold activation over baseline. And then we see a similar result with the MCF7 CRISPR-A cell line. Again, these are the CT curves for VEGFA, EGFR, and HBG. And we also see extremely robust fold activation for all of these genes. We actually see anywhere from 15 up to 1,000-fold activation in this cell line. Okay, so now let's talk about preparation. So what should you keep in, what types of steps do you need to take before you undertake a CRISPR activation experiment? Well, number one, we recommend determining the DNA sequence of the sgRNA target in your cell line. In this case, the sgRNA target site is going to be a gene promoter. Now, this is not essential, but we recommend it because you could design an sgRNA sequence to target a promoter region using the reference sequences for human or mouse or rat. But your particular cell line or sample might have a polymorphism. Now, CRISPR sgRNAs can target some level of mismatches, but it's still best to have 100% match. So this would be necessary to ensure the highest possible nucleotide match before sgRNA synthesis. So if you're going to do this kind of sequencing, we recommend using our Smart Join Blend-In PCR cloning kit to help you with the sequencing. So in this kit is a convenient method for high efficiency cloning of blend-in PCR products, because you're going to want to, what you would want to do is you would want to create a PCR product from your cell line of the region containing your target site, and then subclone that into a bacterial vector. Another advantage of this kit is that it has a higher positive ligation rate and true positive colonies due to the elimination of self-ligating products. And the cloning, it allows for the cloning of blend-in PCR products without additional sequences or restriction sites, and it's ideal for sequencing regions of genomic DNA. This is the workflow for the Smart Join Blend-In PCR cloning kit. We start out with an optional purification of the PCR product. This is, of course, not necessary. And then you clone the PCR product into a blunt end vector, transform bacteria with the ligation product, screen for positive colonies, and again, the positive colony formation rate is well over 99%. So it eliminates any kind of old-fashioned blue-white screening. And then you just do sequence verification. Okay, the next step in the workflow for using CRISPR activation that we recommend is to validate sgRNA function in your cell line. Again, you don't have to do this, but it's a really good idea because not all sgRNAs are created equal. So it's best practice to try multiple target sites and pre-validate the best one. So the way we recommend doing validation is to do qPCR. And if you're going to do that, you can use our GeneCopoeia's BlazeTaq CyberGreen qPCR system. So these are some of the features of our CyberGreen qPCR system. This is it's available as either a two-step system where you would do reverse transcription of your RNA into cDNA separately from the qPCR, and so you can purchase a reverse transcription kit from us, as well as the BlazeTaq qPCR master mix. Or we just recently introduced a one-step kit which does the RT and qPCR in the same reaction. It's highly sensitive. It detects as little as 5 copies of DNA template or 0.1 picogram of RNA template. And it provides high specificity with minimal level of primer-dimer and non-specific primer product formation due to the fact that this is a BlazeTaq enzyme is a hot start polymerase. And this is an antibody-modified polymerase, which provides faster heat activation than chemically modified enzyme. So it requires only 30 seconds of heat activation versus 15 minutes for the older chemically modified enzymes. So this shows some of the performance data of the BlazeTaq CyberGreen qPCR. This is data from the two-step enzyme mix. The amplification plot on the left shows the wide dynamic range of this enzyme, where we can detect, get efficient amplification from anywhere from 5 times 10 to the 7th copies down to 5 copies of DNA. And then the CT curves on the right show performance comparison against a leading competitor, where the GeneCopoeia BlazeTaq enzyme curves are in red and the competitor enzymes are in blue. And we see more robust amplification for the BlazeTaq compared with the competitor. We see similar performance superiority with the new BlazeTac CyberGreen one-step qPCR system. Shown here is, hang on one second. Shown here on the left is a sensitivity measurement where we see better sensitivity with the BlazeTac shown in red versus the competitor, better sensitivity and more robust amplification, as well as this is for two different templates. This is GapDH on the left and Actin on the right. And in the next slide, we see a specificity measurement where again compared with another competitor, and we found that the competitor shows more non-specific amplification compared with the BlazeTac product. Okay, so now finally let's talk about screening. So let's say you're doing a CRISPR activation experiment and you want to screen. You've transfected your CRISPR activation reagents into your cell line. Well, what do you do next? Well, you're going to want to screen for these, and we recommend doing single clone isolation, post-transfection or transduction. And there are several different ways of doing single clone isolation. You can do a classical method of plating for single colonies and picking them off a dish using a cloning cylinder, or if you have the benefit of a fluorescent reporter on the plasmid, you could do fluorescent sorting, which is easier. Or if you're using a suspension cell line, if you're unlucky enough and you have a suspension cell line without a fluorescent reporter, then you would do serial dilutions and multi-well plates. But the bottom line is it's very important to do this single clone isolation because it minimizes potential effects of unwanted modifications resulting from random insertions that you can get from plasmids or cell division or off-targeting. And then finally, to screen, again, you're going to screen clones using qPCR. And I don't need to go through it again, but we recommend screening your single clones to test them for activation using our BlazeTac CyberGreen qPCR kits to analyze them for transcriptional activation. Okay, so with that, I would just like to summarize. I told you that CRISPR-Cas9 mediated gene activation is a useful system for potentially overexpressing genes and has distinct advantages over the use of ORF clones, especially for high throughput screening. Currently, the most robust CRISPR activation system in use is the dCas9 system of Konerman et al., which I mentioned is a three-component system consisting of dead Cas9 VP64, the MSH2 fusion, and the aptamer-modified sgRNA. And finally, I told you that GeneCopoeia offers many products and services, including plasmid DNA clones, Cas9 activation cell lines, and robust qPCR reagents to help you meet your CRISPR gene activation needs. Okay, so with that, I'm going to stop and take questions. Okay, so the first question I have here is, can I increase expression of my gene if it is already highly expressed? That's an interesting question. The answer is it kind of depends. I mean, it really depends on what you mean by highly expressed, but the answer is basically it's certainly possible. It just depends on a lot on the biology of your system. Like, you know, if the protein you're overexpressing is toxic, for example, then you probably would not be able to get much overexpression at all. But yeah, it should be possible. I don't see why not. Okay, next question. I got here late and missed the beginning. Will you be giving this webinar again? We might be giving this webinar again, but even better, we will be making this webinar available as an on-demand recording sometime in the next day or two, so you can always catch it again and again and again. Okay, so next question. Also, the slides will be available too. That people ask that a lot. Next question is it possible to turn on 3 genes at once? Yes, actually, that's a great question. The one of the advantages of CRISPR is it's highly amenable to multiplexing, so you can readily activate many genes at once. So I don't even know if there is a limit. It's probably limited by the number of constructs you can get into a single cell. So 3 should be no problem. It just depends on what the genes are. Okay, next question. Will this work in flies? Yeah, I don't know. To my knowledge, it has not been tested in other systems, although I could be wrong about that. But it should, in principle, work in flies, except that I don't know if the VP64 and MSH2 will function in Drosophila or other organisms like C. elegans, because those are mammalian proteins. I mean, VP64 is a viral protein, but it's from herpes simplex virus, which is a mammalian virus. So it might work, but I can't give you a definite answer. Okay, next question. Why were the gene activations in your MCF7 cell line higher than in the 293 cells? Yeah, that's a good question. We don't really know the answer to that yet. There could be many factors. There could be position effects of the insertions for the DCAS9 VP64 and the MSH insertions, or there could be lower baseline transcription levels of the genes that we tested in the MCF7 cell lines. So that would give an apparent higher activation. But we don't know. But the really important thing is it was pretty robust in both cell lines. So, okay, next question. Are insertion sites in cell hero cell lines known, as those insertions may have their own effects on cell function? Yeah, that's a good question. They're not known. They could be. We used lentivirus, which, as you know, is pretty much random, but we did not determine the insertion sites. So as you point out, and as I just mentioned, there could be position effects. That's why we functionally validate these cell lines before we release them, because we want to make sure they do give robust activation. Okay, so are there any more questions? Anybody? Okay, well, if not, again, I'd like to thank you very much for attending the webinar, and I hope it was useful. And with that, I want to say have a great day. Oh, and please contact us. You can contact me at 866-360-9531, extension 227. Also, my email address is edavis@genecopia.com. And please visit us on the web at www.genecopia.com. All right, with that, take care and have a great day.